Imagine, if you will, a master lockpicker. For centuries, this particular thief has really only ever practiced their craft on one highly specific, incredibly unique type of door.
Right, they're completely specialized.
Exactly. They are an absolute master at breaking into the houses of ducks, geese, and, other aquatic birds. Their tools are just perfectly calibrated for those specific locks.
But not for ours.
Yeah, precisely. If they try to use those same tools on a human's front door, they usually just, well, scratch the paint and bounce right off.
Which is incredibly fortunate for us, honestly, because human architecture is fundamentally different at the microscopic level. I mean, the locks just don't match up.
But what happens when that specialized lockpicker suddenly decides they want to get inside your house? And worse, what if they start hanging around your neighborhood, figuring out how to upgrade their tools to do it?
That is the million-dollar question right now.
It really is. So if you are wondering why we are talking about avian home invasion on Monday, March 23, 2026, welcome to today's deep dive. Today, we are talking directly to you about a phrase you have undoubtedly seen flashing across your news feeds: bird flu, or, as the scientific community calls it, avian influenza.
It's a topic that carries a lot of historical weight and, well, looking at the latest data, a fair bit of current anxiety. The virus is moving into spaces we haven't historically seen it occupy.
Which is exactly why we've gathered a massive stack of sources to figure out what is actually going on. We are pulling from the official clinical guidelines from the CDC and the World Health Organization, incredibly detailed medical reviews from Stat Pearls and Veterinary World, and some cutting-edge virology papers from the JIA Journal.
It's quite the reading list.
Oh, it's dense. But our mission today is to cut through all that noise and shortcut you to being perfectly well-informed. We are going to look at the history of these viruses, the mechanical lock and key of how a duck virus actually jumps into a human, how doctors differentiate it from, like, your average winter cold, and exactly how you can protect yourself.
And I am thrilled to dive into this with you. What's so fascinating here is that this particular virus isn't just a static bug waiting to be swatted. It's an evolving puzzle.
Yeah, and to help connect all these complex clinical dots, I'm joined by our resident expert.
Glad to be here. To really understand the threat profile today, we have to rewind the clock a bit and look at where this virus comes from and physically how it manages to break the species barrier in the first place.
Right, because we hear bird flu thrown around today as if it's, a brand new phenomenon, but the veterinary literature and our stack says otherwise. People have known about this for a long time.
Oh, a very long time. I mean, the first description of what we now classify as highly pathogenic avian influenza was recorded way back in 1878 in Italy.
Wow, 1878.
Yeah. At the time, they just called it foul plague. For decades, it was strictly an agricultural and ecological issue. Even when there was a massive pivotal outbreak at a Scottish chicken farm in 1959, it remained an animal problem.
So the virus pretty much stayed in its lane?
Exactly. Humans were the collateral damage only in the economic sense. Farmers lost their flocks, but they weren't getting sick themselves.
Until 1997 in Hong Kong, that seems to be the chapter where the story completely changes.
That is the turning point.
Yeah.
That permanently woke up the global public health community. That was the year the H5N1 strain caused a terrifying outbreak where it jumped directly from infected poultry to humans.
Yeah, the numbers were really bad, right?
They were. We saw 18 confirmed human cases and, tragically, 6 deaths. That mortality rate just sent shockwaves through the virology world. It proved that the lockpicker could, in fact, get through human doors under the right circumstances.
Okay, let's unpack this, because this is the part that always trips me up when I'm reading these news reports. The sources mention a massive shift starting in 2021 with a strain called clade 2.3.4.4b.
Right, the current dominant strain.
Yeah. And suddenly, by spring 2024, the virus is jumping into U.S. dairy cattle. How does a virus mechanically designed to infect a water bird figure out how to infect a cow?
Well, if we connect this to the bigger picture, it's really about environmental saturation. In mid-2021, that specific variant became incredibly fit, meaning it adapted beautifully to wild bird populations without immediately killing them.
So they were just flying around spreading it everywhere.
Exactly. It became truly endemic globally, spreading across continents via migratory flyways. Because it was everywhere in the wild, the environmental viral load just skyrocketed.
Making it a numbers game.
Exactly. Migratory birds were landing near farms, shedding the virus in their feces, which then contaminated shared water troughs or cattle feed. So it wasn't necessarily that the virus mutated to specifically target cows, it's that the cows were suddenly eating and drinking massive amounts of the virus.
Ah, okay. It's a mechanical transfer. The cows are just living in a newly contaminated environment. But that still leaves the biological question. How does it physically infect the cells of a mammal, whether that's a cow or, say, a human dairy worker? What is the actual mechanism?
It all comes down to the surface proteins of the virus. When we say H5N1, those letters actually stand for the two primary tools the virus uses. The H stands for hemagglutinin.
Hemagglutinin. Got it.
Think of the hemagglutinin as the lockpick we talked about earlier. This specific lockpick is chemically designed to bind to a very specific receptor, a literal molecular lock on the surface of a host cell.
Okay, so the H is the tool to break in, and what's the lock it's looking for?
That lock is known as an alpha-2-spero-3-linked sialic acid receptor.
Alpha-2 color 3. Okay, so where are those locks usually found?
In aquatic birds, those alpha-2-3 locks are incredibly abundant in their gastrointestinal tracts. That is why the virus spreads so easily through bird droppings. It's replicating by the millions in their guts.
That makes sense. But what about us? We aren't birds.
Well, humans do have those exact same alpha-2-3 locks, but nature didn't put them in our noses or our throats.
I assume that's why we aren't all getting bird flu every time a pigeon flies over us in the park. The locks aren't on the front door. Where are they hiding?
They are buried deep, deep down in our lower respiratory tract, specifically in the terminal bronchi and the alveoli, which are the tiny, delicate air sacs at the very bottom of your lungs where oxygen exchange happens.
Deep in the lungs. Okay.
And interestingly enough, we also have a high concentration of those alpha-2-3 receptors in our eyes.
Wait, our eyes. So the lockpicker is searching our house for this one specific lock, and they only find it if they go all the way down into the basement of our lungs or if they land directly on our eyes.
That captures the dynamic perfectly.
That means it requires a massive dose of the virus to make it all the way down there without our upper respiratory defenses catching it first.
It does. It takes profound exposure to reach those deep receptors. But once the virus does bind to those alveoli in the deep lungs, the consequences are devastating.
Because it's attacking our oxygen supply.
Exactly. Because it attacks the very cells responsible for absorbing oxygen, it triggers a massive, sometimes excessive immune response. Your body rushes inflammatory cells and fluid to the lungs to fight the virus, which leads to diffuse alveolar damage.
Meaning the lungs fill up with fluid.
Right. The tiny air sacs essentially fill up with fluid and blood.
Which explains the grim statistics in the Stat Pearls review, a mortality rate of over 50% for human cases of H5N1. I mean, the virus isn't wasting time giving you the sniffles if it gets in, it's immediately compromising your ability to breathe.
It leads rapidly to severe viral pneumonia and acute respiratory distress syndrome, or ARDS. Your lungs simply stop functioning as gas exchange organs.
Wow. Okay, so if the virus needs to reach the deepest parts of the lungs or land directly in the eyes to take hold, that completely redefines what an exposure risk actually is. We aren't talking about catching this from someone coughing on the subway.
Not at all. Human-to-human transmission is currently incredibly rare for that exact reason. You simply don't exhale enough virus from your deep lungs to easily infect someone else's deep lungs.
So who is actually at risk here?
The true high-risk zones are places where there is a massive environmental viral load. We're talking about live bird markets, poultry slaughterhouses, or situations involving direct contact with infected feces, saliva, or feathers.
Places with huge concentrations of animals.
Right. A worker cleaning out a heavily contaminated, dusty chicken coop is inhaling massive amounts of aerosolized viral particles straight into their lower respiratory tract.
And the sources also heavily emphasize the new risk vector from the 2024 dairy cow outbreaks, unpasteurized raw milk.
Yes, this is a critical development. When the virus infects dairy cattle, it replicates heavily in the mammary glands. The udder essentially becomes a viral factory.
Oh, wow. So the milk itself is contaminated.
Heavily contaminated. The CDC guidelines explicitly note that raw milk from infected cows contains staggering quantities of the virus. Drinking raw milk or even a farm worker getting splashed in the face with it during the milking process presents a massive risk of exposure.
But pasteurization fixes that.
Completely. Choosing pasteurized milk completely eliminates that specific threat because the heat of pasteurization destroys the virus's lipid envelope and denatures that hemagglutinin lockpick.
Good to know. So if you are a listener who works in agriculture, or let's say you absolutely had to visit an affected farm or a live bird market, how do you protect yourself? If the virus is in the dust, the feathers, the milk, what is the defense?
The CDC's personal protective equipment guidelines for these environments are rigorous. Throwing on a standard surgical paper mask is insufficient.
So no blue paper masks.
No. You need a NIOSH-approved N95 respirator, ideally one that has been fit-tested to your specific face shape to filter out those tiny viral particles before they can be drawn deep into the alveoli. You also need fluid-resistant coveralls, gloves, and boots that can be thoroughly disinfected.
You know, there was one detail in the CDC guidance that jumped out at me: the eye protection. They specify unvented or indirectly vented safety goggles. Standard safety glasses, like the kind you wear in a wood shop, aren't enough.
Standard safety glasses have open gaps around the edges. If a worker is power washing a contaminated milking parlor, aerosolized fluids or infected dust can easily float through those gaps and land directly on the conjunctiva of the eye.
Where we just established those vulnerable alpha-2/3 receptors are waiting.
Exactly. Unvented goggles create a seal against the skin.
It's like weatherproofing the one exposed window on the house. You have to seal the frame, not just put up a screen.
That's a great way to look at it. And this concept of interrupting the virus extends to systemic public health strategies, too.
Like what?
Well, in regions that rely heavily on live bird markets, authorities institute mandatory resting days. They completely empty the market of all live birds for a day and sanitize the entire area.
Oh, I see. To break the chain.
Yes. It breaks the chain of continuous viral replication that happens when unsold infected birds are kept caged next to new healthy birds overnight.
That makes a lot of sense. But let's look at the clinical side. Say the weatherproofing fails, or someone is exposed unknowingly because they didn't realize the backyard flock they were handling was sick. What happens next?
It's a critical window.
The Stat Pearls review mentions early symptoms are just fever, cough, and muscle aches. I mean, that sounds like a Tuesday in January for most people. How do doctors actually spot the difference between bird flu and a really bad winter cold?
This raises an important question about clinical vigilance. The early signs can be incredibly deceiving, but the timeline and progression are huge tells.
How so?
Seasonal flu primarily hits your upper respiratory tract, has a short incubation period of about 1 to 4 days, and is generally self-limiting. Avian flu, however, can take up to 10 days to incubate after exposure.
Up to 10 days.
Wow. And when the symptoms do escalate, they progress astonishingly fast to severe dyspnea, which is profound shortness of breath.
My mind instantly goes to COVID. If I wake up coughing and struggling to breathe, I'm assuming it's that. How does a doctor know it's not?
A key differentiator is that avian flu does not cause the hallmark loss of taste or smell that is so strongly associated with COVID-19.
Okay, what about bacterial pneumonia? If a patient is coughing up a lung and running a high fever, how do they rule out a bacterial infection?
With bacterial pneumonia, a patient will typically produce purulent sputum, meaning they are coughing up thick, pus-filled mucus.
Yikes. Okay.
Furthermore, a chest X-ray will often show lobar consolidation. That means an entire distinct section or lobe of the lung fills up with fluid, almost like a water balloon. Avian flu, being a viral pneumonia, does not typically produce that purulent sputum, and the imaging looks completely different.
Here's where it gets really interesting, though. Because there is a telltale sign that seems completely unrelated to your lungs. We talked about those receptors in the eyes earlier.
Conjunctivitis, severe eye redness, tearing, and inflammation.
Yes. The sources noted that this was the predominant symptom in the recent U.S. dairy worker cases, which ties the whole biological lock-and-key concept together perfectly.
It really does.
The workers were getting splashed with contaminated raw milk, the virus found the alpha-2/3 receptors in their eyes, and boom, conjunctivitis.
It is a textbook example of pathophysiology dictating clinical symptoms. And it's exactly why doctors are now specifically trained to ask about occupational exposure if a patient presents with a combination of severe respiratory distress and red eyes.
So let's follow that patient. They walk into a rural clinic, they have the red eyes, they are struggling to breathe, and they mention they work on a dairy farm. The medical system has to instantly switch into high gear. Why can't they just use one of those rapid flu swabs we all have in our medicine cabinets to confirm it?
Oh, using a rapid antigen test for suspected avian influenza is a massive risk.
Why is that?
Those rapid tests are calibrated to detect the specific antigens of seasonal upper respiratory flu strains. Because bird flu replicates deep in the lungs, the viral load in the upper airway where you are swabbing the nose or throat is often too low for the rapid test to catch.
So it just misses it.
The medical literature shows rapid tests frequently result in false negatives for H5N1,
So a false negative could send a highly contagious, critically ill patient right back into the waiting room, assuming they just have a mild bug.
which is a catastrophic failure of infection control. Instead, the absolute gold standard for confirmation is RT-PCR testing, specifically assays designed to detect the H5 and H7 genetic subtypes.
Because PCR actually amplifies the genetic material, right? It's looking for the needle in the haystack and copying it until it's visible.
Exactly. RT-PCR is highly sensitive. It can amplify even tiny fragments of viral RNA from a deep respiratory sample, and it can yield definitive results within hours.
Okay, so while the lab is running that PCR test, the doctors are also doing imaging. You mentioned earlier that bird flu doesn't look like the lobar consolidation of bacterial pneumonia. What does it look like?
Doctors will use CT scans to get a highly detailed cross-section of the lungs. Normally, on a CT scan, healthy lungs full of air look pitch black. But with avian influenza, doctors are looking for diffuse ground glass opacities.
Ground glass, like a frosted bathroom window.
Precisely. Instead of healthy black voids, the scan shows hazy gray frosted patches spread diffusely throughout both lungs.
Meaning the fluid is everywhere, not just in one lobe.
Right. That gray haze is the visual representation of the alveoli filling up with inflammatory fluid and cellular debris. It is the hallmark visual of acute respiratory distress syndrome ravaging the tissue.
So if you see that frosted glass look on the scan and the PCR comes back positive, what is the treatment protocol? We talked about the H and H5N1 being the lockpick. Where does the N come in?
This is where we target our clinical defense. The N stands for neuraminidase.
Neuraminidase.
Yes. If the hemagglutinin is the tool the virus uses to break into the host cell, the neuraminidase is the tool it uses to break out of the host cell once it is replicated so it can go infect neighboring cells.
It's the getaway vehicle.
It is. So the primary antiviral medications used to treat this are neuraminidase inhibitors, like oseltamivir.
So they block the exit.
By inhibiting the N protein, the medication effectively traps the newly replicated viruses attached to the dying host cell, halting the spread of the infection through the lungs.
But the sources stress that timing is everything with these inhibitors.
Time is absolutely critical. They need to be administered as early as possible, ideally within 48 hours of symptom onset.
And if they aren't,
The virus replicates so aggressively that if you wait too long, trapping the virus won't matter. The immune-mediated damage to the lungs might already be irreversible. At that point, the patient will require mechanical ventilation or even ECMO, extracorporeal membrane oxygenation.
wow, ECMO. Where an external machine has to take over the work of the lungs entirely by pumping and oxygenating the patient's blood outside their body.
Exactly. It's a last resort.
That is an incredibly intense intervention, and obviously if hospital has a patient exhaling a virus with a 50% mortality rate, they have to lock that down immediately to protect the healthcare workers.
Clinical infection control is paramount. Hospitals institute standard, contact, and airborne precautions simultaneously. The patient is immediately placed in an airborne infection isolation room.
With negative pressure.
Yes, these rooms use negative air pressure, meaning air is constantly pulled into the room and filtered rather than flowing out into the hospital hallways. And every healthcare worker entering that room is wearing the full, rigorous PPE we discussed earlier, the fit-tested N95s, gowns, gloves, and unvented eye protection.
It's a massive reactive defensive operation. But what about proactive defense? I mean, millions of people get their seasonal flu shots every autumn. Does that offer any cross-protection against this?
Unfortunately, no. Seasonal flu vaccines are calibrated specifically for the human strains currently circulating, the ones that bind to the upper respiratory tract. They offer zero protection against avian H5N1.
But the StatPearl source did mention that bird flu vaccines exist. Why aren't we all rolling up our sleeves for those?
Specific vaccines targeting H5N1 and other avian strains have indeed been developed and are maintained in national stockpiles. However, they aren't distributed to the general public because the risk of everyday exposure is incredibly low.
So who gets them?
They are used strategically in a process called ring vaccination.
Ring vaccination, like building a wall around the fire.
Yes, exactly. If a major outbreak occurs at a poultry facility, public health officials deploy these specific vaccines to the high-risk populations, the poultry workers, the outbreak responders calling the flocks, and the frontline healthcare providers treating the patients.
So you heavily protect the people on the absolute front lines to sever the chain of transmission.
Exactly. It prevents the virus from gaining a foothold in the broader human community.
So what does this all mean for you, the listener? Let's summarize the key takeaways. Bird flu isn't just a sudden new panic. It's an ancient virus from wild birds that is constantly learning new tricks, like contaminating shared environments to jump into dairy cattle.
But it has limits.
Right. Right now, its biology is its own biggest limitation. It relies on a very specific molecular lock and key, the alpha-2 best-free receptor. Because humans only have those locks deep in our lungs or in our eyes, it requires significant direct exposure to infected animals or raw milk to get sick.
Exactly.
Which is why unvented gobbles, N95 respirators, and completely avoiding unpasteurized raw milk are your absolute best bets if you are ever in a high-risk agricultural environment.
It is a disease defined by exposure.
Yeah.
If you manage the environmental exposure, you successfully manage the risk.
But as we wrap up, I know you found one final detail in the virology papers regarding the genetic structure of this virus, a detail that looks to the future, and frankly, it's a bit unsettling.
It is something the global scientific community watches very closely. As we established today, human-to-human transmission is currently rare because this virus prefers the alpha-2 receptors buried deep in our lungs.
Right.
But here's the catch. Unlike some viruses that slowly mutate over time, influenza viruses have a segmented RNA genome. Think of their genetic code like a deck of cards.
Okay, a deck of cards.
If a pig or a human gets infected by an avian flu virus and a normal seasonal human flu virus at the exact same time, those viruses can shuffle their decks of cards together inside the host cell. They can deal out a completely new hybrid mutant virus practically overnight. This is called reassortment.
Wow, so they just swap traits.
Exactly. If the H5N1 virus undergoes a reassortment or mutation that allows its hemagglutinin proteins to efficiently bind to the alpha-2 receptors, which are the locks overwhelmingly abundant in the human upper respiratory tract, in our noses and throats, the entire game changes.
Because then it's in the easy-to-reach places.
Right. If that receptor switch happens, it wouldn't just be an occupational hazard for farmers and dairy workers anymore. It could theoretically spread through a simple cough or sneeze on a subway, just as easily as the common cold, but carrying the severe pathophysiology of avian influenza.
That master lockpicker finally shuffling the deck and finding the master key to the front door, that is definitely something to mull over the next time you see a flock of wild geese fly by. Thank you so much for joining us on this deep dive. We hope you walk away feeling informed, prepared, and ready to share this knowledge. Stay curious, stay safe, and we will catch you on the next deep dive.